computer in 1950s developed what is called the Coulter principle. What is it? Basically the same thing. You have an electrolyte solution and you have another area in which vacuum pump is applied so that all these particles which are there in the electrolyte solution should pass through the aperture into the pump area or a separate tube. Each cell, when it passes through that aperture, gives rise to a pulse because it is an insulator and is causing defect in the current pathway. And this has been used for the evaluation of these parameters and has led to marked improvement in the reproducibility of the blood cell counters, and now these parameters can be used to the advantage of the patient. Next please. And there are certain other parameters which are the ones which help us whenever we are looking at these counters. So these counters give us all these parameters. Some are measured and some are derived parameters, and they generate RBC, WBC and platelet histograms. Now what are histograms? They are blood counts which are performed by machines, can have a VCS technology, what is called the volume, conductivity and scattered, what I have shown you just now, or it may be multi-angle polarized scatter separation. Certain high-end machines use that. Now, what is the limitation in terms of morphology by these counters? These counters cannot recognize polychromatophils, stippling, a lot of red cell parameters cannot be seen by these machines. The WBC also, number of parameters like atypical lymphocytes, Pelger-Hugh, toxic granules, band forms cannot be seen by these machines. Aggregations cannot be seen. Leukoerythroblastic blood picture, panocytes, Roulet, all these cannot be seen by these counters, but still they have advantage. And we must appreciate that and keep these things which cannot be seen on the machines. So you have to look at them. Although some of the higher end machines these days are giving you immature granulocytes, nucleated RBCs, toxic changes, but yet most of us are using 3-part differentials which are incapable of giving that. Regarding hemoglobin estimation by these counters, we know there may be a limitation of hemoglobin, can be falsely elevated whenever the leukocytosis is more than 1 lakh, or maybe severely lipemic serum, abnormal protein circulated. And please remember, it may, although very, very little percentage, less than 2% is sulf, and choleglobin, but it will not measure these. Please, certain parameters are required. If you're getting a very falsely high hemoglobin due to certain plasma factors, many people use the plasma replacement or plasma blank facility in these counters. Next. So what is a histogram? As I showed you, whenever a cell passing through an aperture, it causes a peak or a disturbance in the conductivity. And these signals are generated, which are according to the size of the cell. And then these signals are analyzed by the computer in our machines, saying that this is the size likely to be WBC, platelet or RBC. So histograms are produced for these 3 parameters. That is how the histogram is generated for WBCs. We know they are from 50 to 400 femtoliters. So according to the size, the machine will put the smallest, the lymphocytes on the left side, increasing in size, coming to the monocytes and the eosinophils in the middle, that's the mid population, and then the neutrophils on the right side. That is what a blood smear looks like graphically. That is what the machine is looking at. Next, it has generated so many pulses. Each pulse is a cell. Next, and then it starts analyzing these by putting them into some order according to the size and generates a beautiful curve which is the maximum number of cells falling in this region, and it analyzes that according to the size of the cell, and that is what is a histogram. Again, we know platelets are usually 22. You can see 2 to 25 femtoliters. There is a, what we call the discriminators are put below that platelet, above that RBC, above that WBC. Next, please. Same thing, you can see that they are peaks in different regions according to the size, because there are certain